It's my pleasure, honor to introduce Dr. Erik Henricson. He's a friend. He's one of the first persons I say hello when I came to the US more than 23 years ago. And now we're saying how old we are. And yeah, right. And he is also at UC Davis, and he is an associate professor in the Physical Medicine and Rehabilitation Department. He's also a co-director of the research lab. He has been doing amazing things in terms of mobility, investigation, outcome measures, even like going all the way to learn more about the cognition in patients with Duchenne muscular dystrophy. So again, I'm eager to hear your talk now, Erik, and thank you again for being here.
All right. So yeah, thanks very much, Carolina, for the introduction. Yeah, we've been doing this for, it seems like forever now, and I'm really delighted and kind of amazed to see the progress that collectively the DMD and neuromuscular worlds have made in the last 20 years, and it doesn't diminish at all the challenges that we have today. But I'm happy to say that we're at a point now where we at least know a lot more about how to measure different aspects of the lived experience of DMD, be that all the way down at the molecular genetics level of gene mutations and cellular changes, all the way up through looking at time strength and time motor performance, and now we need to start thinking, as well as we're testing different interventions, about how do we know that people are feeling better, right? And so those are the things that we need to start looking at for personal factors and environmental factors, and how people are interacting with their environment and in the context of society. And so I'm actually a human development guy, in addition to being a public health guy and sort of a computer science guy. I wear a lot of hats, but it makes it fun because we get a chance to really look from a multidisciplinary standpoint at different aspects of what it means to have DMD, and how can we kind of put this all together. And so this is kind of my research focus, which is a little bit of a beautiful mind kind of thing. But realistically, if you start here on the left and look at the primary changes in muscle and muscle cells, and then muscle tissue, and then muscle as an organ, right? That are due to dystrophinopathy, so we're just going to call it that, Duchenne, Becker together, how that relates to muscle strength, and then how that then leads to changes or in ability or mobility of a person. And that could be mobility of skeletal muscle, upper extremities, lower extremities. That could be mobility of the diaphragm and breathing and chest wall. So mobility takes on a lot of different definitions, and we can think about things that go with that, like fatigue or pain or sleep disturbances. These are all things that then, if you think about the last time you got a really, not the greatest night of sleep, and then had to go and perform highly someplace, the next day, that impacts your behavior, right? It certainly impacts mine. Just ask my wife. The cognition and your ability to kind of participate in daily activities, right? So these are the things that we need to start to think about. And so when we meet up with folks at the FDA, for instance, and they say, okay, well, you're showing that you're making somebody go a little faster, right? In their walking in the community, 95th percentile stride velocity is going up. How do you know that people feel better? And that's the question that I'm really interested in, right? So how do we do that? Well, the first thing I'm going to tell you a couple of different projects that I have going on. One is this one that Carolina and Craig and I have been doing with our colleagues with the Synergy Network for the last 20-odd years. And then I'll tell you a little bit more about person-reported outcomes, and I'll tell you a little bit more about wearable devices and how we can look at community mobility, and by that meaning not just walking, but actually just getting around, and how that impacts your resources you're able to access and things you're able to participate in. So, first act, Duchenne Natural History Study, right? This is 20 years in the making. And when we started this out, of course, we all looked exactly the same as we do today. But I think it's easy to just think about natural history data as something that's there, that's always been there. It's just, it's a data repository. But what I have to tell y'all is that this is the work of literally thousands of people for thousands of hours globally to put this together, right? And I'm not going to, everybody knows this in this room, right? That we have a multi-system developmental disorder, right? We've got impact on skeletal muscle, but then we've got lungs and heart and GI tract and bone and also brain, right? And so that's one of the things I'm not really going to talk about today, but just remember one of the things, as we look at this natural history study data, one of the things that we've done in clinical trials so far is that we've typically said, we want folks who are reliable in strength and function measurements. That cuts out a portion of our population who may have more of the kind of developmental disorder kind of looking cognitive challenges, and so probably weakens our data just a little bit. But we need to think about how to address that, right? So this starts out, the story of synergy, right, which was the idea that we get folks together worldwide to start looking at this DMD is a rare disease because it was, with a couple of exceptions, the CIDD group, or being the main one, it was a disorder that had been looked at as single center studies. And because of that, and I have to highlight that, so Craig did a single center natural history study that if anybody in the room, is not aware of, I'm not buttering him up. It's fantastic. It served as a model for everything that we're doing here. And with, 150 people at UC Davis over several years, and as a part of a larger profiles in people with neuromuscular disease project. But it really laid a lot of the groundwork for what we're doing here. And then layering that on top of the Synergy network, where we got overwhelming response from folks saying, we want to participate in multi-center studies. We want to try to be just like the National Cancer Institute of the muscular dystrophy world. We were really lucky to get all of these collaborators globally. And this led to, we started off doing some small clinical trials, and we kind of learned as we went along because we didn't have a whole lot of expertise in the neuromuscular world in clinical trials. So we drew from as many people as we could, but we were able to put the system together that was really highlighted as this new NIH roadmap concept that they had around the early 2000s of having multidisciplinary groups work on projects that were too large for one specific discipline. And so we were lucky to be kind of highlighted, by the director of NIH at the time, and but it really does. It helped to. It was nice that the NIH and other agencies were starting to really look at how we were putting multidisciplinary teams together, and that led to this natural history study being this group of folks here worldwide, and this is just our PIs who were working on this, and our advisors. We were in 9 nations at 24 different hospitals, and we enrolled 440. We often say people with DMD, but realistically, we enrolled the families, and that means triple that at least, right? In terms of the number of participants, more like 1,200, and if you add in siblings, it goes up from there. And we, over the course of 12 years, we had more than 3,500 visits, with these multidisciplinary teams, and we had people from 2 to 28 years old. We had people who were ambulatory, non-ambulatory, who were on varying degrees of steroid therapy. And remember, when we started in 2005, 2006, and actually doing the bulk of these assessments, steroid therapy was not universally accepted as the standard of care. So we actually had a lot of folks who were steroid naive as well. The schedule, and this, you start to see these time frames mirrored in clinical trials that we see today. We had short-term changes of the every 3 months because we wanted to see not only retest reliability, which we had established for some of the measures earlier on, but what are the short-term changes that we can see every 3 months, for instance, in different age groups with XYZ, whatever the assessment set was that we were using. And then how does that change in the longer term? And we were able to, in many cases with a lot of our participants, follow them for anywhere from 4 to 10 years. Over the long term. So that gave us some very powerful data in terms of looking at not only the longitudinal progress of loss of or change in function of the disease, but how that also related to individual characteristics, whether they be genetic or age-based or treatment-based, or even the age cohorts of when they entered the healthcare environment. And then this is where this multidisciplinary team came in, is we have the H&Ps, but medication reviews, strength and function, demographics, quality of life, and looking at this whole set of different topic areas and data domains. So when you look at the data, list of data in the data dictionary for this, it's like 4,000 variables or something like that for every single measure, or every single visit, pardon me, and it covered all of these areas, including cardiac care, pulmonary care, musculoskeletal stuff. And that was kind of where we were, if I can say that we were innovating a little bit, it was we were combining these disciplines, right? And we got people together around the table, and people said, I want to add this, and I want to add this. And we did, and it was hard, but it, in most cases, turned out to be useful. So when we think about kind of the outputs, right, they always want us in academia to quantify outputs of projects. This is kind of, it's not a victory lap, right? But this is evidence that this kind of multidisciplinary, multi-center model of looking at, the boring part of natural history that doesn't involve clinical trials, it doesn't involve, direct intervention, is still really useful. We've got 17, I think it's more like 18 or 19 novel drug therapy groups that have used this data so far. We've got papers on modifier genes, biomarkers, modeling and prediction of natural history, validation of clinical trial outcome measures, as well as papers on pulmonary health, cardiac health, a little bit on neurodevelopment. I've got some work to do there. I really got to start cranking on if I ever get time to do it. And quality of life, right? So we're starting to fill in the gaps and starting to draw parallels here between these different domains. So this is what it looked like. And I know many of you are at least familiar with Erik Hoffman, who was our boss at the time. And we started this up in 2005. And then we just spent time in the trenches collecting data. And when I say we, I mean the families as well. So not just us, not just our clinical research coordinators, not just our clinical evaluators and PTs, but everybody was working on this. And see all the publications that happened here in the first 5 years? Yeah, that's what Hoffman came to my desk one day and said, you are spending a whole lot of money doing this, and you've used a whole lot of resources, and I don't see a single paper out of this. You are committing academic suicide here. What are you doing? And it was hard to argue with that a little bit, and I said, nope, this is a public health project. Public health projects take a really long time. Just wait. And he went, and he walked out. Great, okay. And once we got enough, that's this whole, we felt great about right here. But once we developed enough data to start asking the right questions and to start answering the right questions, then it sort of took off, right? And you can see just from the kind of avalanche of things that happened after that, we were able to start building and building and building and working not just within our group, but also with pharma sponsors, with investigators from other institutions worldwide. And so this is kind of what the original natural history study has built into today, right? And it's a fairly widely used global data set. And so, just a couple of key findings. We're able to show with the first paper that that Craig and I did with all of our synergy investigators was that one of the things that we were able to show was the benefit of steroid therapy for people maintaining upper extremity function, because it was typically the case that when somebody stopped walking, they would discontinue steroids because we don't want them to be exposed to any of the health risks of steroid therapy after that. And, some of us thought, well, it's probably still got some benefit there. And we were able to show that that was the case. And it moved the needle a little bit on clinical care. And this is kind of the interesting and powerful part about these kinds of studies, right? You can start to look at different groups, steroid-treated, non-steroid-treated, how different durations of steroid therapy might lead to future preservation of function or lack of loss of function. And we were able to, look at that over time, over a great number of folks who were, in the steroid-treated or non-steroid-treated groups. And this has again moved the needle on utility of steroid therapy, and I think overcome a lot of people's, not the concerns exactly, but at least reduce the questions of whether are we just poisoning people, right? Which is the word that a lot of folks used when we started to have this discussion back 20 years ago. Now today, what we're seeing, instead of that kind of descriptive stuff, we're seeing much more use of our synergy data and data from other sets worldwide, right, as historical control information for clinical trials. So a sponsor will come and say, well, okay, we just did this clinical trial. Our study population looks like this. Do you have anybody in your study population who looks similar to that? Can we see how they did over a similar period of time? And this is, this has been really helpful, actually. And so we've been able to show that compared to, for instance, the natural history group, which included steroid-treated folks as well, that the a group of eteplirsen-treated patients actually had a delay in their progression of the worsening of their pulmonary function over time, relative to folks who were measured within our earlier cohort. Same kind of thing here. Stabilization of functional mobility over a longer 2-year period of time in the Vidal-Larsen study was also something that we were able to show by developing a synergy data cohort that could be used as a comparison in addition to the cohorts that they had in the original design. So, when you look at the overall impact of the project so far, we've got more than 1,100 scientific citations. It's mostly basic research articles touching on, hey, there's DMD and kind of talking about the potentials for intervention. But we have also clinical practice articles that are, like, for instance, like the ones with the steroid utilization that are starting to change the standards of care for DMD. Those include things like the ATS guidelines for pulmonary management, the cardiac management guidelines with the American Heart Association, steroid use guidelines with the American Academy of Neurology. We have multiple drug approvals now. I keep updating the slide and it keeps being wrong, right? Because we've got, it's a good place to be. The Centers for Disease Control had, looked to some of our earlier study outputs and conclusions when looking at the DMD care considerations project. And most recently, I found some interesting things. The government of Brazil has a care consensus statement now that referenced our study. The Blue Cross Blue Shield folks are looking at some of our published information from before so that they can determine both their coverage guidelines as well as their treatment response guidelines. We have a ways to go with that, but at least they're starting to look in the right places. So this is the NASCAR slide, right? I wish I had a lab coat with all the little, it's kind of like the race car drivers. But it does highlight, the one thing that it kind of gets left off here, and MDA is on here, and PPMD is off here, but on here, but the individual families, if you had pictures of everybody, it would fill the whole screen. But it is really a global effort, and it's not only a global effort in the academic world, but it's a public-private partnership. And this is something that everybody from the NIH on down is trying to encourage now. So where are we today? 5 countries, 10 hospitals with our new, what we're calling the expanded Duchenne Natural History Study, which is essentially a reboot, right, of the original one that we did starting in 2004, but with a more contemporary cohort of participants. And right now, we're looking at, so Sarepta was our initial funding group and has jumped in with a tranche of funding. We're still looking for partners, whether it's through NIH grants or other pharma industry folks. But the idea is, and this is not a new model, is that this is research in what they call pre-competitive space. This is the kind of information that everybody needs to move the field forward. And this kind of model of having multiple sponsors come in and collaborate and sit around the table to achieve the goal of developing a tool or developing a resource that everybody can use is something that's used in the seed, in agriculture and seed development. And it's used at UC Davis, we have a strawberry disease consortium, and we have an electric car battery consortium that's done with Mercedes and Ford and Chrysler, everybody together. So we're not inventing this. But what we have is essentially the same model as what we had before, with a partly new group of players, some previous, some of the sites that were part of the original study as well. We're in the process of getting that launched and getting contracts signed and things, so I can't quite put it all on the screen yet, but it's happening. And by the end of the year, we're going to have all this together, and we're doing the sort of we're doing the launch phase right now at UC Davis, because one of the things that you always want to do with the new study is have a couple people who are patient go through it and figure out where you need to tweak it to make it run the best. It's kind of like adjusting the carbs on your engine. You got to make sure it's all adjusted right. The specific aims, again, are also very similar. We want to look at changes over time, longitudinal change over 1 to 3 years. We want to look at the changes in the data from clinical outcome measures that are being used in clinical trials today. And we're trying to anticipate as well, what are we going to need tomorrow, right? So because we're doing kind of, those phase 2, early phase 3 to mid phase 3 studies. What do we need for real world evidence studies? What do we need for community mobility, things like that? And we also, then if you think about that earlier question that I had of how do you know that people are feeling better? Well, we need to anchor those changes that we see in those clinical measures to changes in how people report their own function. Not just mobility, but also quality of life, health satisfaction, things like that. So this is this is what our cartoon looks like for this one. We will have evaluations of genetic characteristics. We're going to be collecting samples for blood-based and urine-based biomarkers. We're going to be collecting in-depth medical histories that include medication use and cardiac health and therapeutic use, bone health, GI, lung. We'll be doing assessments of strength and mobility that Dr. McDonald did a fantastic job of giving you an overview of kind of everything that's being used right now. Pulmonary function. One of the things we're adding here that we're really interested in is this neurodevelopmental assessment. So we have a couple of tools that are autism screeners, and you will hear people say that some people with DMD have autism. There's arguments about that. There are common characteristics there. We'll leave it at that, but we have more to learn about people with DMD and how dystrophin in the brain changes, affects behavior and development, and neural development as well. So, and then finally, some tools looking at quality of life. And when we say quality of life in this context, it's both self-reported mobility, which isn't really quality of life, it's just mobility. And then actual things that involve life satisfaction and health satisfaction and things like that. This is a list of the outcome measures. People always ask, like, oh, you know, these are going to be really long visits. Yeah, they are. And, you know, realistically, you know, when we have folks come in for these kinds of studies, it's a hard sell sometimes. And this is why we're trying to point out to people what the benefits of developing these kind of data sets actually are. Because when you come in to a clinic to participate in one of our natural history studies, there's homework for the parents doing person-reported outcomes. There's homework for the guys doing person-reported outcomes. There's a variety of different age-based or functional ability-based assessments that, again, were things that Dr. McDonald had talked about, and we're adding in like the Bailey Infant Development Assessments because we don't know enough about babies. So if we're able to get babies in the door, we want to get a good picture of what they look like from early development through early childhood, where we feel much better about knowing what the functional abilities are of guys with DMD, right? And we also have the socio-emotional evaluations, questionnaires about developmental ability, things like that. So we have one set of outcome measures that's really for ambulatory participants, and then we have another set of outcome measures, there's a lot of overlap, that are also age-based, that are for individuals who are non-ambulatory. So it gets to be kind of a complex web to look at and see who does what at what visit based on their age and based on their ability. But suffice to say, it's not everything that's here. But it's enough that it starts to let us kind of color in that. Craig had a slide that showed the different assessments over time and by age and by stage. We have to fill in. We don't have the luxury of being, like, you know, one of the clinical trials where we're focused on one group of people, so we pick one outcome measure. We have to be able to pick all of them, essentially, and figure out a way to convince everybody to work with us. And we've been super, super lucky with this. People have said, yep, we'll do it. And we've had kids who have come back year after year after year to do these things. So this is the researcher cast of thousands. You know, we have a huge number of collaborators. And so this is, you know, truly just the sort of quintessential team science project outside of like particle physics and super colliders and things like that. This is really cool. So that's where I'm going to stop about the natural history study. Actually, I'm on time. That's rare. Does anybody have any questions about that part before I go on? Let everybody take a little break.
I have one question. So how, I saw where you wanted to get people of different races and ethnicities involved.
Yes.
How are you going to get to the people who can't get there?
That's a great question. So this is a big challenge. One of the things that we're very lucky in that many of our centers are regional centers where, you know, folks do unfortunately travel a very long distance to get where they're, you know, going to come see us. It does create barriers, and it's something that we're very aware of. One of the things that we are, you know, I'll talk a little bit about the community wearables part. There's some sort of telehealth, sort of mobile technology things that we can start to look at in that context. The other thing is that there's like the MDA Mover program that is collecting maybe not as in-depth data, but allows us an area to compare to, because we always want to be able to kind of go and check our data against other centers. It might not be that there's exactly the same, but there are points that we can look at to try to decide whether we're similar to everybody else or whether we might be a little bit off target. And one of the examples that we were just talking about earlier today is that, so for our first study, we had most of our sites around the world were in the developed world, for lack of a better term. And then we have one site where they have thoroughly modern healthcare in southern India, but it's a developing nation, and some of the folks who were coming there are coming from 500 miles away in really rural areas. And what it turned out was that a lot of our guys from the population in India looked very different. And at first we're like, oh, well, that's a problem. And as we've looked at it more and more, it's actually a strength. And so one of the things that we are trying to do now is make sure that as we are a global project, that we are not a biased global project where we're looking entirely at the developed world.
Well, even in California, we have migrant family workers
Yes.
who are not being.
And that is definitely a challenge as well. And it's one of the reasons that we have spent extensive time and resources translating all of our materials into Spanish, for instance, and translating into multiple dialects of not just Hindi, but Tamil, in different regions, and in Israel, and who is on our list to start up, and they're in a pause right now. Translating into both Hebrew and Arabic, because we do have folks both in Jerusalem, West Bank, from both ethnic groups. So we're not going to perfectly solve this problem, but it's an ongoing. It's actually a discussion point in our regular meetings. Are we doing a fair enough job? Because the reason for this is that we want to make sure that, especially with everybody using our data, we don't want to have that challenge that the data that we're gathering turns out to be not be multi-ethnic and not apply to the rest of the world. As the other parts of the world wait to get these new therapies in and on board, we need to make sure that the data that we're using for sake of comparison is actually informative to treating folks in those parts of the world as well. So stay tuned. We're trying. Sir.
Thank you, Dr. Henricson. Looking at the recent result from Embark by Sarepta, the primary endpoint from the North Star result was not really significant, while at the same time, all the secondary endpoints are significant, indicating that improvement is really not captured. And also Dr. McDonald suggests, talking about the decline usually doesn't happen in early age patients. So are you looking into ways of improving the North Stars and how this is going to be used in the future clinical trial, and would that also affect how you build this global database in the future to be more accurately capturing all the changes?
That's a great question, and so we don't directly have input on the content or the design of many of the individual evaluations. What we do have, however, is I talked about this concept of anchoring, which is essentially just kind of knowing like where A and B link up. When we have data from multiple tools in the same domain, let's just say muscle strength, for instance, and you would expect them to behave in similar manner over time. Different tools have different floor effects below which they don't do a great job of measuring, or have ceiling effects above which they don't do a great job of measuring. And as we move through the age groups of people with DMD and we're using different tools, those ceiling and floor effects can vary. So we need to be able to line those up as best we can. It helps us develop a continuum of measures that represent that one domain, but it also helps us to understand if we see that one tool moves, say, in an upper range and another tool does not, we can go, oh, well, okay, so everybody who's, let's say, scoring 34 on the North Star could have gone higher over here, and we just lost that because 34 was as high as you could go. Those kinds of things. So it gives us at least a lens through which we can look to see whether the measurement ranges for the tool are sufficient for the population of participants who we're looking at, which is another part of it and kind of why we're trying to look at these broadened age ranges and broadened levels of ability as well.
Eric, if I could just make one comment.
Go for it.
Just real quick. But, with regard to the North Star, in having discussions with some of the investigators that have been sort of pioneering the development of the North Star, they actually added the time to rise and the 10-meter walk are actually parts of the North Star. So actually there were essentially, and that's because of these ceiling effects and the lack of granularity. So in a sense, some of those components of the North Star actually did have statistical significance. They just weren't pre-specified as the primary endpoint. Eugenia McCurry has actually even been talking about, well, maybe we should make modifications to the North Star with regard to items such as the running item. Because if you can get both feet off the ground at the same time, you get a score of 2. Well, that's not normal running necessarily. There might be a lot of additional improvement that can take place, but you still only get a score of 2 on the North Star. So we've been talking about maybe even components of the 10-meter walk run test would sort of translate to a score of 3 or a 4 on the running. So there have been discussions about ways we can sort of tweak the North Star. But I think any clinical trial done in that young 4 to 7-year-old population has just generally not been, unless it's a steroid-naive population, looking at the effect of steroids, the North Star just hasn't been as sensitive.
So your question was perfectly lined up then with that question of responsiveness is part of what you're talking about. Is are we when we see a change and we measure change, is it a real change? And if we measure no changes, there are truly no changes. There's something going on that we're simply not measuring. And so I'm looking at this from a little bit of a different angle in looking at changes in self-reported mobility. Now remember, not everybody, but a lot of folks throw the person-reported outcomes. If it's a questionnaire, it's quality of life. Well, yeah, okay, but not really. And so one of the things that we have seen, and folks assumed, even many of us made kind of an assumption, mobility equals happy, right? And that we would see that happiness and life satisfaction kind of things would go down with declining mobility and ability to walk and hand to mouth and things like that. Well, we see the very clear differences in transferabilities, basic mobility over increasing age, and we also see that in upper extremity function. When you look at happiness scores and some of the tools that we were using, at the same time, what do you see here? It doesn't change that much. It's not what we sort of expected when we started to talk to the FDA about, well, we're measuring that mobility and people can report those different. And they said, well, are people feeling better? And we go, well, of course they're feeling better. Well, you could argue looking at this that maybe it's not changing that much. So why is that? Maybe it's complicated, right? It doesn't happen in a bubble, but it also means that when we're doing clinical trials, we can't make the assumption that being more functional equals being better. Obviously, there's a part of that that's true. But if you look at how the context of people in their lived daily lives, maybe it's more like this, right? Strength is related to mobility, relates to participation in some way, which then relates to quality of life, right? And so things that affect a person's participation, right? That could be strength, it could be fatigue, could be mobility, health, behavior, but it could be family resources, things like that. So, we can think about reduced participation, but we can also think about things that create increased participation despite the fact that we have reduced mobility, right? So there are, I'm going to skip over the behavior part, but this kind of goes back to this idea of kind of pulling out the quality of life part of that earlier diagram that I was showing Here are things that we think are probably related, but the relationship between strength and happy, right? This is all the things that are in between. And moving the needle on any of these things could affect happiness and quality of life. So this is what's called a bioecological systems theory model. There's a whole group of behaviorists and child development folks that have worked on this since the 1970s. And so I'm liberally lifting from them in these sort of concepts. But if you look at the challenges and impact of somebody with DMD on family life, right? You get increased physical stress and emotional stress, and there's sorrow and worry and financial stress and things like that. But there are also supportive things as well. And so we can think about everybody in this family system contributing to the wellness of the participant, and the participant contributing to the wellness of the family, right? So you think about siblings. What are impacts for siblings, and what are impacts on the extended family? Many people, as I've talked to them, say they have challenges with talking to folks, their parents, grandparents, and talking about not only kind of behavioral expectations for kids with DMD, but also physical abilities and school participation, things like that. So involving extended family in supporting roles, we've heard that theme a couple of times today, is probably beneficial in some way as well. So you have these positive potential for positive reinforcing factors that are multiple links, right? People sharing and talking about their feelings and worries and focusing on the wellness of the family. This builds family resilience. This is especially in the face of physical and medical challenges. But many people also report that they develop closer and more meaningful relationships, and they're able to start to look at what matters the most in life, or they have, identify, a spirituality that they hadn't realized before, or that the family can share in the search of resources. And you think about this, and then the family is nested within your community, and you have the same kind of potential for interactions with members of your employers, with healthcare resources, with resources at schools, recreational groups, activities, and then at the state, national, global level, you have governments, you have advocacy organizations, and when you think about how all of these things intermix and realize that nobody is completely in their own little bubble, we have to start looking at the support web. When you talk to folks who are in early phase clinical trials, and this is in many life-threatening conditions, not dystrophinopathies specifically, but when you ask them about what are the benefits of being in a clinical trial, you get a lot of folks who say, well, I know that this phase 1 clinical trial for my pancreatic cancer isn't really going to help me, but maybe it's going to move things forward for somebody else in the future. There are these ideas of sort of beneficence and trying to help the community, and that makes people feel good. So it's relative. One of the things that we also need to think about for clinical trials and what makes, how do we get to happy, is that we have things that change over time. The needs of a family of a 2-year-old or a 4-year-old or a 5-year-old are going to be very different than the needs of a family of a teen who's, somebody who's in high school, or someone who's graduated from high school and is looking at college or is trying to move into the working world. So the players in what determines happiness and what determines life satisfaction are going to be vastly different for those different groups. So when we start to look at that, what seems like a simple question from the regulatory folks, do they feel better? Showing that somebody is stronger does not necessarily mean they're going to be happier. There are lots of happy, weak people. There are also a lot of really unhappy, strong people, right? So you have to think about, and that could just be what that your milk was sour for your breakfast cereal in the morning. It can be these little things, right? So these are the kinds of things that we need to think about and start to measure. that we haven't really in the past. I'm going to talk very briefly about the lifetime mobility scale, which is basically this idea of anchoring again, which is one of the things that we've done with the data from our natural history study was we compiled that and started to look at the questions that we asked people to answer from a variety of different tools about their own functional ability. We asked parents about their children's functional ability. We asked kids and adults with DMD about their own functional ability. And we got a set of questions that we felt represented a range of function from really strong, right? All the way through, just some of the guys who Craig was showing who have, some hand function, but, can't get their hands to their mouths, things like that. And so the idea here is that, when we say we've shown that somebody is 20% stronger on their, their bicep strength, many folks in the regulatory world will say, okay, so what? Show me that 20% increase actually was something that patients could perceive, right? That they felt like not only could perceive, but felt was important to them. And so as we look through all of these different questions, what we've assembled for what we're calling the lifetime mobility scale is a set of lower extremity walking and moving questions, trunk stability and positioning changes, whether it's getting in and out of a chair, rolling yourself over in bed, things like that, and then upper extremity function, hair combing, toothbrushing, scratch your nose, sit and eat a meal, those kinds of things, and characterizing how the changes in function that people report correlates with the changes in function that we see in all of these clinical tests, because that helps us in a number of different ways. One is it helps us, creates a continuum of measures, right? Because that slide that Craig was showing earlier, we have time motor performance tests, and then we have pulmonary function, we have 6-minute walking. Many of those tests work in a certain range of people, right? But we're actually talking about functional ability. So this gives us a tool that allows us to look over the entire range of functional ability as one number. But it also, we're talking about tasks like, how easy or hard is it for you to brush your teeth, or to comb your hair, or to get up and down out of a chair? It's tasks that are daily tasks that a 10-meter run walk doesn't really tell you about. And so we can start to use those to interpret changes if we know that changes in walking velocity are also related to self-reported changes in walking in the community. We can actually start to say, okay, well, we know that going a half a meter per second faster actually means that you're probably likely to be able to also go up and down your curbs in the community, okay? So it helps us to interpret and develop a richer story around what are otherwise, in-clinic laboratory-based assessments that are, kind of dry. They're very responsive and they're very finely, they're finely resolute, but they don't really tell us what people are like in the real world. So, which gets me to the community mobility part. And bear with me, because somebody's gonna say, well, you're just talking about walking and there's lots more about mobility, but I'll get to that part. One of the things that we want to do, right? So Craig was talking about 95th percentile stride velocity. This is a great tool, actually, because it's letting us then start to look at how fast people are moving on a kind of a usual basis in the community. But there are things that it still doesn't tell us, right? So one of the things we're trying to do with our wearables Walk For Me project is develop cost-effective or maybe free ways to measure community-based gait, but also mobility, and to tie that into some of the habitual behaviors that go around, like where do you go? Not just that you can walk at such and such a speed, but where do you do it, and does that matter? And so we had to develop some tools that let us do kind of build individual gait fingerprints for people using some AI technologies, especially for folks who have atypical, or disordered gaits, your average like iPhone doesn't cut it with the software that's in there. And then we developed some machine learning based tools to help us learn more. And so, these common tools, I'm not going to really go over because Craig talked about them, but the clinic is not a real world setting is kind of the take home here. We want to understand not only what people are able to do, but what they do with it. And so this is actually part of a study that Craig did, again, as a part of his original natural history, but looking at the stopwatch activity monitors back at sort of when they were first being developed in the late 1990s, early 2000s. And they're great at looking at quantifying steps per day, but also step frequency and at minute by minute intervals, which lets you look at sort of low frequency steps, medium frequency steps, and high frequency steps, which gives you an idea of like how fast you're trying to get someplace. But one of the things that we found out as we tried to launch this in clinical trials is that the steps per day in people with DMD on the balance for most of the steps actually didn't change that much, which is why that 95th percentile of the fast steps actually matters. And so now we have some more specialized devices that are allowing people, they not only count steps and step frequency, but are developing these temporospatial characteristics of stride length and stride velocity. This is really important, right? Because it lets us know not just about the kind of the timing of the walking, but how is somebody walking. That said, they're still really expensive, right? Some of the lab grade tools, $10,000 per year per person to do these measurements. In the context of a clinical trial, maybe if you're a drug company, that's okay. But for little old us with the public health project, no way, not for 440 families for 12 years. We've looked at some of the consumer level devices, right? Everybody's seen the Garmin's and the Fitbits and all the fancy tools for runners and things like that. Well, they are not very accurate in people with disordered gaits because a lot of these are developed for the athletic community. They're designed to track how fast you run and how fast you did your last marathon. Not really useful for somebody who's walking at 1 meter per second and most of their gait is like side to side motion. Okay, so what do we do about this? We have to develop new ways to do things using simple available tools, right? So can we measure steps in people with weak gaits using cell phones? Well, it turns out actually your mobile phone sensors are the same as the sensors that are in these lab grade tools. They're pretty much identical. And if you can get to the data and you can convince somebody to wear one for long enough on the little belts around their waist like all the runners do, right? Turns out you can actually do a pretty good job of teaching the computers to identify the gait patterns for individuals, right? You do a little bit of calibration, say this is what a person's slow walk, typical walk, fast walk, and a jog or a run look like. And you say, okay, this is the signals for this. And they're using machine learning techniques. The computer can then go, oh, wow, this is a slow walk. And not only that, but by observing it and watching how fast people do a 10-meter run walk, you can actually then use those signals to teach the computer what the stride lengths are for each of those, which means that you can start to replicate those lab grade tools, but using your iPhone and an app that you download. And so that's kind of where we are with that right now. And the accuracy of these tools, if you look at just the step counter in your iPhone, the one that you look at in your iPhone health and says, oh, look, you went 1,500 steps today. Sort of, maybe. They're about 60% accurate, right? For typical walkers. They're really, now if you're really hoofing it, it'll do a better job of you're moving and hoofing it across campus. It'll count that okay, but your little steps around the house, things like that, doesn't count them at all. So we need those kinds of things because a lot of what the folks with neuromuscular diseases outside of DMD, with DMD, whatever, people take smaller steps. They take gentler steps. And they may take just the same number, but it may not get them as far. So we need to be able to measure those, right? So using a variety of different machine learning techniques, actually our outputs are a little bit better now. We can actually not only measure distances with about 96 to 98% accuracy, so step counts and distances traveled, but then when you feed this in and say, okay, does this data set that you've just seen, Mr. Computer, does this look like data from a typically developing person or from somebody with DMD? Our models now for that are like 95% accurate. And slap a cell phone on somebody, have them go walk for somewhere between 1 to 2 minutes is 90% accurate down to toddlers. Not all toddlers, we're getting there. We still we have far to go in terms of getting data, but returning a result of this is somebody with a typical gait or this is somebody with a disordered gait. So getting there. So what do we do with that? It's very computationally intensive, right? And from a clinical trial standpoint, maybe not, maybe it doesn't have that much utility. We'll see. But when we look at community gait, we want to understand more about how people move in the community, not just how fast, but about the behavior itself. So we're learning. These are the kind of signals. You look at this blue and red signal. This is what your iPhone will give you if we turn on the sensors and you strap it on and go for a walk. This was me walking around the park. This is not new either. We're stealing from the bird songs and musicians and doing signal characteristics here. We can start to see what patterns of activity look like for longer periods of time, and we can visualize it, right? So this map down here at the bottom, that was not just a couple of steps. That was about 25, 30 minutes of activity, just walking around the park. So again, not new. This is the Loma Prieta earthquake. This looks like a similar signal, right? And so we can transform that into looking at frequency. Essentially, walking, your walking, my walking, anybody's walking, is a repetitive signal. It's got a frequency and it's got an intensity and a magnitude. And so you start to use those words and you type those into Google, you come up with earthquakes. And so what we're thinking about is, can we start to use this kind of sensor data on a much larger, longer-term scale to generate maps to look at how people are able to move in the community. If we're able to do this and understand how people can move around and how effectively they're able to move around, whether it's speed or intensity or step count, we can learn about behavior. And that gets us closer to the happy part. So this is a little experiment we did in lab using the sensors with a super slow walk, kind of a typical walk, and then progressing onto adding in a fast walk, which is where you start to see that purple band at the bottom, and then a jog, just on a treadmill, and adding in a minute or two of jog until you got up to 4 minutes. And you can see that the different kinds of activity are visually very easy to pick out. So this down here, that's running. This is a fast walk. This is kind of a slow, comfortable walk. And you can see this for any group of participants. So if we look here, here's a control participant. We did a bunch of different exercises out on the track. 6-minute walk, 25-meter walk, 100-meter run walk, and some free walk. And here's this person actually did, they did the 6-minute walk here at the fastest possible, but they decided for their free walk, we gave this woman instructions to walk slowly, or just to walk comfortably. She decided to run. She was so happy to run. You can see that signal very nicely here.
Sorry, we have like a few minutes.
A few minutes, okay. So anyway, the long and the short of it, if we look at somebody with DMD, we don't see the same kind of pattern. So we know that getting to something that looks more like a typical level of effort over time is a desirable thing. we can also map this, and we can start to look at where people were doing, like, for instance, a little jog right here, and you can map that out in terms of velocity. So we're getting to the point of starting to look at gait characteristics and mobility and participation. This is our next step, and this is where we really think it's going to be interesting for folks, is looking at multi-day data to look at the habitual activity that's associated with mobility. If we can do that, which has been done in other disorders, then maybe we can draw the lines between mobility, geographic location, and happy. And that's it.